In this lesson, we're going to look at biological molecules. So the learning outcome is the first one, describe ring forms of the alpha and beta glucose. Number two, define the term monomer, polymer, macromolecule, and monosaccharide, and disaccharide and polysaccharide. Number three, covalent bonds in joining smaller molecules to form polymer. Number four, glucose, fructose, and maltose are reducing sugars, but sucrose is not. Number five, describe the formation of glycosidic bond by condensation in disaccharides and polysaccharides. Number six, describe the breakage of glycosidic bond by hydrolysis in disaccharides and polysaccharides. Refer to non-reducing sugar tests. Now, these are the basic terms that you need to know when you study about biological molecules. So this has been covered in your secondary school, so I will not touch on it. Please revise on your own. Now, the chemical on Earth, all chemical compound on Earth can be divided into two types. The first one is the inorganic compound, and the second one is the organic compound. Now, inorganic compound, they are found in rocks and mineral deposits. For example, sodium ion, potassium ion, magnesium, gold, silver, and so on. Whereas organic compound, they are formed by organisms. So all organic compound on Earth, or all organic molecules on Earth, they are all produced by living things or organism. So carbon is the basic element, and usually they will have carbon, hydrogen, oxygen, and sometimes nitrogen, phosphorus, and sulfur atoms. Now the carbon atoms are joined by covalent bond. Now, why covalent bond? You see this carbon atom, they are joined by this covalent bond here. So this is covalent bond, right? So why covalent bond? Because covalent bond is a strong bond. So it gives stability to the overall molecule. So the carbon atoms are joined by the covalent bond into either chain form or ring form. So this one is a chain form and this one is a ring form. Now, the chains and the rings both are the carbon skeleton of organic molecule. So the backbone of organic molecule is either in chain form or in ring form. Now, the term monomer. Monomer is a relatively simple molecule which is used as the building block for the synthesis of polymer. So that means the building block is just like the little bricks, right, that we use to make the wall. So the simplest unit to make the polymer is the monomer. So it's the basic building block for the synthesis of polymer. So we join n number of monomers together to form the polymer. So usually monomers are joined by condensation reaction. Now the word condensation in English means water droplets are formed. Now, in the case of chemistry, condensation means that when two molecules join together, water is released as part of the product. So condensation, so water becomes one of the product. So in the diagram, you can see this is one unit of monomer, which is the simplest building block. And when you join all of them together, it will form a polymer. Now, what about polymer? Polymer is a giant molecule. It is made up of many similar repeating subunits. So the monomers becomes similar repeating subunits. So all of them are similar and they repeat itself. Just like the pearl, right? You string all the pearl together. So each of them is a pearl. So the similar repeating units along the chain of the polymer. So they're all joined together. Monomers are joined together in a chain. Now, monomers are smaller and simpler molecule. Now, when you come across the word macromolecule, macro stands for big, molecule is a molecule. So macromolecule is a very large biological molecule, which is an organic molecule. Okay. For example, polysaccharide, protein, and nucleic acid. So these three are classified as macromolecule. Now, test yourself. Which of the components labeled A, B, C, and D is or are the monomer? Now, a monomer must be a repeating unit, right? So this is a one unit here, and this whole unit repeat itself. So this is the monomer, right? The whole unit repeat itself. So here in the table, I'm showing you the four major group of organic molecules that you're going to learn in your syllabus. So the first one is carbohydrate, followed by lipids, followed by protein, and lastly, nucleic acid. Now, carbohydrate, the basic unit of the monomer is called monosaccharide. Right? Mono stands for one, sacchar stands for sugar. So one unit of the monomer. Then the monomers are joined up by glycosidic bond, which is a type of covalent bond, to form polysaccharide. Then lipid generally is made up of fatty acids and glycerol. So fatty acids and glycerols, they are joined by ester bond. Lipid does not reach the macromolecule level because lipid does not have many repeating subunits. It's only made up of 3 fatty acids and 1 glycerol. So there are no repeating subunits of 3 fatty acids glycerol, which repeat itself again and again and again in a long chain. So therefore, lipid does not reach the macromolecule level. Protein is made of amino acid as the monomer, joined by peptide bond to form the polypeptide. Nucleic acid is made of nucleotide as the monomer, joined by phosphodiester bond to form the nucleic acid, for example, DNA and RNA. So we will first look at carbohydrate as the biological molecule in our body. Carbohydrate is a molecule which contains carbon, hydrogen, and oxygen atoms, whereby the ratio of H2O is always 2 to 1. So 2 to 1 is same as the ratio of water, right? Because water is 2H and 1O, so similar to water. So the general formula for a carbohydrate is CxH2OY. CxH2OY. Now, the word carbohydrate, hydrate actually stands for water. So carbohydrate is a hydrate of carbon. So based on the formula, you can see that it's like a water together with carbon. So it's like you're adding water to carbon. So it's hydrate of carbon. So carbohydrate. So let us look at sugar. Under carbohydrate, we've got sugar. Sugars are either monosaccharides or disaccharides. That means you've got either one sugar unit or 2 sugar units. Sacchar stands for sugar, right? So all sugars and sweet are sweet and soluble. So for example of the sugars are triose. Triose means a 3-carbon sugar. For all sugars have got the suffix end with O's, O as is. So the suffix for sugar is always O's. So you got triose means 3-carbon sugar, tetrose means 4-carbon, pentose means 5-carbon, and hexose means 6-carbon sugar. Now, polysaccharides. Polysaccharides, they are not sweet and they are insoluble. So only sugar is soluble. Polysaccharides are not soluble. For example, starch, glycogen, and cellulose are polysaccharides. They are all not soluble. So here you're going to look at the glucose as an example of the monomer of carbohydrate. So we're going to learn the glucose structure as an example of monosaccharide. So over here, what you can see is, this is a structural formula of a glucose. So this is a chain form of glucose. Now it is made up of 6 carbon atoms. Each carbon atom from carbon number 2 to carbon number 6. So this is carbon number 2 until carbon number 6, right? So each of them has got a H and an OH attached to it. Now carbon 1 is this one here. Carbon 1 is part of the functional group of the glucose. It's a functional group of the glucose. So what is the functional group? All organic molecules have their own functional group. Now, functional group is a specific group of atoms within an organic molecule that gives the molecule its specific property. Like, why is it this is alcohol? All right. Why that one is ketone, aldehyde? Why is it carboxylic acid? So it gives the property to the molecule. So in this case here, the C-double-O gives the property, C-double-O bond gives the specific property to this sugar molecule. So therefore, the carbon atom is numbered starting from number 1 for the carbon that's nearest to the functional group, right? Carbon nearest to the functional group will be numbered as number 1. So that's why you can see the numbering here is number 1 here, right? Followed by number 2, number 3, number 4, number 5, and number 6. So that's how we number the carbon in any organic molecule. Okay, so ignore the number first. So from a chain form of glucose, it can also form the ring form of glucose by rearranging itself, right? So what you can see here is, can you see the O of carbon number 5 will join with C of carbon number 1 in order to form the ring form of glucose. So therefore, when a ring form is formed, the red color carbon is carbon number 1. So this carbon number 1 is joined to the pink color O, all right, which belongs to carbon number 5. So therefore, in this ring form of glucose, we have got carbon number 1 here, followed by carbon number 2, okay, followed by carbon number 3 here, then carbon number 4 from here, carbon number 5 from here. Now, because this part here form a ring, so therefore this carbon will be outside the ring. Carbon number 6 will therefore be outside the ring. So therefore it's shown here that carbon number 6 is found outside the ring here. So this is carbon number 6. So altogether still got 6 carbon, but one of the carbon is outside the ring, because even though this ring is hexagonal, but one of the corner is occupied by the O. So therefore the 6th ring is outside the ring. So here I'm showing you the chain form as well as the ring form of a glucose molecule. Right, so this is a chain form of glucose molecule, can rearrange itself into the ring form, and there are 2 ring forms for glucose molecule, either alpha glucose and beta glucose. Now, can you see the differences between alpha glucose and beta glucose? You check the carbon number 1 here. You see the OH that is attached to carbon number 1. This OH is pointing downwards from carbon number 1. Whereas this OH for beta glucose is pointing upwards. So the differences between alpha and beta glucose is the OH on carbon number 1 is pointing downwards, whereas for beta glucose, the OH for carbon number 1 is pointing upwards. So how do we remember this pointing upwards and downwards? Very easy. Now, when you write the word alpha, you write it this way. So it goes down, right? So therefore pointing downwards. But when you write beta, beta is actually written this way. So it's pointing upwards. So up and down. Very easy to remember. Now, when you want to write the carbon ring form, Either you write it in this way that's shown here, right? That means every carbon that you have, now, can you see this is hexagonal shape? All right. Every corner is a carbon. So in organic molecular formula, organic molecule structural formula, every corner that's occupied by carbon, you don't have to write. It's understood it's a carbon. Now, do you see this O has to be written because this O here is not a carbon. It occupies the corner of the molecule. So we have to write O there because it's not a carbon. Now, for the rest of them, we don't write because it's carbon. So the moment it's carbon at the corner, we don't write. Now, for every corner of the carbon, there's one OH and one H attached to it. So therefore, when you draw the ring form of glucose, you need to include all the H and OH attached to each carbon. So therefore, you need to memorize the location of the OH. So you can usually memorize this way. OH, OH, H, OH. So I memorize this way. So therefore, all of the elements pointing downwards is OH, OH, H, OH. So therefore, carbon number 3, OH, is pointing upwards because the one below here is H. So the one above is OH. Right? Now, the last carbon pointing outwards, okay, is CH2OH. Okay, so that's how you need to write. And don't forget to also indicate that this is an O here. Now, if you want to make it easier to draw, there's another simpler way to draw this ring form of glucose. Now, this is the one that's also acceptable. So that means to say that I will exclude the carbon, I will exclude the hydrogen, I will include all the OH. So this is another way. So can you see there is a bending here? So every corner that bends, if you don't write anything there, that means there's a carbon here. All right, so this carbon is CH2, right? So CH2OH. Okay, so this is a CH2 here. CH2, that's at this corner, followed by OH, right? Now, another simpler form for you to remember, but not for you to draw, is this, a simpler form. So if you imagine the glucose as a person with 2 hands, and this person wear a little hat over here, okay? And then you got two hands, and both hands are carrying OH. OH here and OH here. Now, because the O is attached to the carbon, so I must write H followed by O. Okay, it's the O that's attached to the carbon. So, and then there is this carbon number 6 up here. So I need to include carbon number 6, and I include the bending. All right, so this is a special antenna on the head. Okay, a special antenna. So this is how you can remember the structure of an alpha glucose. 2 hands are pointing downwards. There's a cap on the head and an antenna on the head. Now, if you want to represent beta glucose, the only difference is that one hand is up, the other hand is down. So that's the only difference. The rest of them are the same. Okay, so this is for you to remember. So glucose molecule can exist in 2 ring forms. The alpha glucose, where the OH of the first carbon points downward, and the beta glucose, where the OH of the first carbon points upwards. Okay, so this is the diagram that you need to draw into your notes. So please draw these 2 diagrams and also the name of the glucose into your notes. Okay, now I'm going to show how we join 2 glucose molecules together to form a disaccharide. And from disaccharide, we join more glucose subunit to form a long chain, which we call them polysaccharide. So monosaccharide plus monosaccharide becomes disaccharide. You add some more monosaccharide to disaccharide, you form the polysaccharide. Okay, so the example we're going to look at here is alpha glucose plus alpha glucose. Okay, so when 2-alpha glucose join together, we will have to remove water molecules from them. So 1 water molecule will be removed when 2 glucose molecules join. So where exactly is the water molecule removed? It is this HO that I circle in red. So water is H2O. So therefore, which part the water comes out from? It comes out from the H of the first glucose molecule and the HO of the second glucose molecule. So it forms HHO. So the process is called condensation because water is released from the process. So therefore, water becomes the product of this process. So water is one of the product. Okay, so minus water. Condensation means you remove the water from your substrate. So oppositely, I can also break down the disaccharide to release 2 monosaccharides again. So the breakdown process is opposite of condensation. So it's called hydrolysis. The word hydrolysis stands for, lysis means to break, right? To break, to lyse. Hydro means water. So hydrolysis means you break the molecule, and then when you break the molecule, you cut the molecule into 2. So let's say, for example, this is disaccharide, right? So you break it into 2. So when you break it in 2, then each of them will have a wound on it. So the part that you cut, there's a wound here. So this wound, okay, this is the wound, and this is the wound. So you need to seal the wound with water. You need to seal the wound with water. So that's why you use the water to seal the wound. One of them, you patch it with a H. The other one, you patch it with OH. So that's why hydrolysis. Okay. Okay, now we want to extend. If from disaccharide, when I want to make it longer, what I'm going to do is that I need to add some more monomer to the right side of my disaccharide. So it is added in one direction only, okay? So I'll add more monomers in one direction. So add one new one here, after that add another one to it, and add another one to it to make the chain longer and longer. So over here, I'm showing you that, okay, in order to add the new monomer unit, it must happen by condensation reaction. So again, water molecule is removed for every unit that's added in. Okay. So water molecule removed is condensation. If you want to break the polysaccharide and make it into disaccharide and monosaccharide, you also have to remove the sugar subunit or the monomer one by one from the end backwards. That means you're going to remove from this one first, okay, the last one, then you remove this one, then you remove this one. So one by one, going inwards, you remove it. So every stage of removal is a hydrolysis reaction. Every stage of removal, you remove one water molecule, right? So therefore, after you remove them by hydrolysis, water, you have to add water to seal the wound. Okay, so polysaccharide can be drawn either in this manner. So this one shows you there is a long chain, and this bond here indicate that this long chain is still connected to more to come. So the chain is actually longer, but you only draw 3 subunits to show that. Or you can represent it in this way. You represent it in this way, that means you got n number of this unit here. So you got an O here, and this O is joined to another covalent bond on the right side of the O. So another one here, right? And then plus water molecule is when you join the monomers together, water molecules are released. So you join n units of this, then you get n number of water molecules being removed. Now there's one more thing I want you to pay attention to in this chart is, can you see this 1,4-glycosidic bond? So once the 2 alpha glucose molecules are joined together, so the water molecules will be removed already here. So this O will be joined to the carbon number 4. So therefore, this is the O here. This O will be joined to carbon number 4. So this whole structure here is that I circle in purple color is called 1,4-glycosidic bond. Okay, 1,4-glycosidic bond. It's actually alpha 1,4-glycosidic bond. Now, why is it called 1,4? It's because a bond that join two sugar subunit together between carbon number 1 of first sugar and carbon number 1 of second sugar subunit. So it's 1,4. And this bond is called glycosidic bond, right? Glycosidic bond has an O in the center, and then you've got a covalent bond that join to two neighboring carbon. So this whole thing here, the O and the covalent bond on both sides, the whole thing circle is called the glycosidic bond. You cannot point to the O because the O is oxygen. You cannot point to this because this line is a covalent bond. So you must circle the whole thing. This is glycosidic bond. Right? Now, alpha here because the two sugar subunit is an alpha glucose. So that's why the glycosidic bond form is called alpha, stands for alpha glucose. 1,4 stands for the two carbon that join together is carbon number 1 and 4, and then glycosidic bond. So let's look at what happened when two alpha glucose molecules joined by condensation. So water molecule be removed, and then now the alpha 1,4-glycosidic bond is formed. Now let us look at the maltose. So once maltose is formed, how do we hydrolyze them? So first we cut them and we seal the wound using water. So this is hydrolysis to give you back the two alpha glucose molecule. Now these are examples of disaccharides. The first one, you've got the glucose plus glucose will give you maltose. Second one is a glucose plus fructose will give you the sucrose. Okay, can you see that the fructose, when you draw it, is in a pentagonal shape, right? Whereas glucose is hexagonal shape. But both of them also got 6 carbon, right? So you see fructose is very interesting, right? It's got 2 carbon that are outside the chain. Can you see there are 2 carbon outside the chain? So that's why the chain is pentose shape, lack of 1 carbon. Okay. Now let us look at polysaccharide. So the learning outcome is number 7, describe the molecular structure of starch and glycogen and relate their structure to their function. Number 8, describe the molecular structure of cellulose and outline how the arrangement of cellulose molecules contributes to the function of a plant cell wall. So there are 3 examples of polysaccharide they're going to learn in your syllabus. Firstly, starch. Secondly, glycogen. Thirdly, cellulose. So in this lesson, we're going to look at starch and glycogen only. Now, starch is a polysaccharide. That means it's made up of polysaccharide chain, right? Okay. So starch can exist in 2 forms. The polysaccharide chain can either exist in the form of amylose or amylopectin. So this is a representation of amylose, a molecule, and this is a representation of amylopectin. Now, amylose is unbranched. That means the polysaccharide chain is unbranched, and the glucose residues are linked by alpha 1,4-glycosidic bond. Now, the moment the individual glucose join up together to form a long polysaccharide chain, each of the glucose inside the polysaccharide chain is called a glucose residue. Okay, the subunit inside the polymer is called a residue. So glucose residues in the amylose, they are all linked by alpha 1,4-glycosidic bond. Now, for amylopectin is branched. So I've shown you here the branch. So this is the main chain here. All right. So this is the main chain. And these 3 are the branched chain joined to the main chain. So these are the branches. They are the polysaccharide chain that form the branches that join to the main chain. So in the main chain, glucose residues are joined by alpha 1,4-glycosidic bond. The branch chain join to the main chain using alpha 1,6 glycosidic bond. So that means to say that at this point here, where the branch chain join to the main chain, okay, so alpha 1,6 glycosidic bond will join the glucose of the branch chain to the glucose residue of the main chain. Okay, so these are the free alpha glucose molecule, which are monomers. So we're showing you how they join up by condensation and release the water to form the polymer. So one by one, they're being joined up. So once the glucose monomer is attached to form the polymer, each of them in the polymer is called a glucose residue. Okay, so this is showing you unbranched chain of the main chain of the amylose. Now, in the three-dimensional structure, the amylose is actually coil shape, right? It's a coil shape. Why is it a coil shape? Because the alpha 1,4 glycosidic bond, which is this bond here, it is not 180 degrees. 180 degrees means when one glucose joins with another glucose, is straight, isn't it? Because this one here stands for 180 degrees. So if the glycosidic bond is 180 degrees, then the entire chain is actually does not coil up, right? But the alpha 1,4 glycosidic bond, they are not 180. They are less than 180 degrees. So therefore, you imagine when one glucose is joined to another glucose, right? It bends a little bit. Then another glucose join up is like this. Another one join up is like this. Another one like this. Okay. Another one here. Then you join another one here. Then join again another one here. So can you see? So you start to coil up this way. Join some more, coil up some more. Okay, and so on. So that's why the whole amylose structure is a coil. And when you do an iodine test to the starch, you add the iodine solution. So the iodine actually sits in the space within the coil of the amylose to form a complex with the amylose. So that complex gives us the blue-black color, right? Blue-black color. Okay, when you add iodine solution to it. Now, what about amylopectin? Now, amylopectin has got branch chain joined to the main chain. So let us look at the branch chain, which location of the branch chain joined to the main chain. Okay. So you see this glucose residue of the branch chain joined to this glucose residue of the main chain. So this one here is carbon number 1. And this one here is carbon number 6. Right? So this carbon number 1 and this carbon number 6. So the glycosidic bond that's formed here is actually 1,6 glycosidic bond. So it's an alpha 1,6 glycosidic bond, which join the branch chain to the main chain. So when all of them are joined up together, this is how amylopectin will look like. Each chain is not straight, it's slightly curved, okay? And then it has got branch chain joined to the main chain. And every branch chain has got another branch chain joined to it. So this is amylopectin. So this is what you need to draw into your notes. So the first one is show the amylose, which is a straight and unbranched chain, okay? When I say straight, I mean unbranched chain, right? Unbranched chain. So now this is the simplest way you can illustrate glucose without paying attention to the rest of the additional OH and H, except for the main one, right? So I'm trying to illustrate here. The glucose residues are joined by alpha 1,4 glycosidic bond. So carbon number 1 joined with carbon number 4 in amylose. So alpha 1,4 glycosidic bond is bent, so the chain becomes spiral or coil like spring. Now, In amylopectin, the structure of amylopectin is similar to glycogen because both of them got main chain and branch chain. So what you can see is that, so on top here is the branch chain joining to the main chain using a alpha 1,6 glycosidic bond, whereas the residues in the main chain, they are joined up by alpha 1,4 glycosidic bond. So the residues along the same chain, they are all joined up by alpha 1,4 glycosidic bond, right? Between the branches, they are joined by alpha 1,6 glycosidic bond. So this is the overall structure of a starch. Very interesting. Okay. Now, what about glycogen? Now, glycogen is similar to starch. The only difference is that glycogen is more highly branched than the amylopectin of the starch. So it's more highly branched. The chains and branches can fold into compact form. So like here. So this is highly branched, right? This is highly branched. So this is example of a glycogen molecule. So in a small space, there are so many branches of the chain that can occupy. So it takes up less space. So the whole structure is very compact. So it takes up less space. Now, glycogen molecules clump together to form granules that you can see in liver cells and muscle cells. So you see on the right side here, this is one liver cell, and this is another liver cell here. And you can see the cytoplasm of this liver cell is filled with the pink color granule, whereas this particular liver cell on the right side, there is no pink color granule. So these pink color granules are the glycogen granules that occupy almost the entire cytoplasm of the cell, because the function of this liver cell here is actually to store glycogen. So that's why it takes up almost the whole space. So why glycogen is used as the storage molecule? We will look at it afterwards. So that's the function of glycogen molecule. Okay, so function of both starch and glycogen is as food and energy storage compound in the organism. Starch is an energy and food storage compound found in plant and seed, whereas glycogen is a storage, energy and food storage compound found in animal and many fungi. So why starch and glycogen are the molecules that are used to store the food and energy is because they are insoluble. So when you store them in the cell, it will not attract water into the cell so that the cell will not swell and burst. Then, because it's insoluble, it will not easily react with any other component inside the cell. Because a lot of reactions in the cell, the chemical reactions, it must be in the form of aqueous. That means it's soluble in order to react with another component inside the cell to form other component. So when the glucose are all converted to starch and glycogen are no longer soluble, so it will not react, so you will not lose them when you store them. Okay, and then glucose is very reactive because it is soluble, so it can react and be converted to a lot of other things like CO2 and water in cellular respiration. It can be converted to lipids. It can be converted to amino acids. Now, these molecules are also compact. That means in a small little space, you can fill it up with a lot of glucose residues. So it's compact. So when it's compact, it occupies less space. So it's a good storage compound. So when you want to keep your books, right, you stack them up together and you tie them up, you put it in the box so it becomes compact. So it takes up less space in your storeroom, so you can store a lot of things. So in the same way, we group up the glucose into chains and we branch them out and we bend them. So in the entire small space, you can store a lot of units of the glucose residues. Okay. Then lastly, starch and glycogen are easily converted back to glucose. Now, whatever that you store, when you want it back, you want it to be easily accessible, right? When you store something in the fridge, whenever you want it, when you open the fridge, you want to see it there. And it's easy to thaw and to be used or to be eaten, okay? You don't want to put in the fridge and you open it, it's gone, okay? So this one, you can easily convert back to glucose by the glucose residues being removed one by one from the outermost unit inwards. Now, branching is very good. You see why? So I'm going to illustrate why branching is good. So let's say, for example, here, when you want to make the glucose residues available again to the cell, you will always remove the glucose one by one from the far end inwards. So we're going to remove this glucose residue at the end of the chain, one by one inwards. So branching will allow more glucose residues to be released per unit time compared if it's not branched. You see, if it's not branched, right, any one time you can only release one glucose residues. But if it's branching, in this case here, at any one time you can get 3 glucose residues being released as glucose molecule, right? So the glucose must be easily converted and made available. Now, this is a past year question. It says that glycogen may be broken down to form glucose. So the question asks the student to draw an annotated diagram in the space provided to explain how a glucose molecule is formed from the free end of the glycogen molecule shown in the figure. Total marks, 3 marks. So that means you need to draw to illustrate how this chain is hydrolyzed so that you end up with one glucose molecule and the chain, which is made of 3 glucose residues. So you need to show that. All right. Okay, I'm going to show how we're going to show that. So first thing is you must look at which is the last glucose residue, or we call it terminal glucose, right? Between the left-hand end and the right-hand end, which glucose residue do you think is the terminal glucose? Right, you always remove from the terminal glucose. So this is the terminal glucose because the OH of this glucose is free, is not bound to anything. So it's not connected to any more glucose residue. But here you see there's a glycosidic bond. So that means it's actually attached to another glucose residue, which is not shown here because it's very long, right? So therefore, hydrolysis must occur to the first glucose residue on the left side, or we call it the terminal glucose residue. So hydrolysis must occur by adding water. So you have to write there, you add water molecule plus H2O. And then this process is called hydrolysis. So you need to indicate the name of the process and what are the molecules involved. And after that, you have to redraw back the glucose molecule. Now, I'm not going to redraw back. I'm just going to show you draw the same thing. And then over here, you will draw a vertical line downwards and put there OH. Okay. And then over here, you draw a vertical line down here after you cut away the glycosidic bond. And here also is an OH. Okay. Take note, the O is attached to the carbon, so OH. All right. And then, so that's all. That's all you need to show. So let us see whether we can score full marks or not based on the mark scheme. I'm trying to remove my drawing, but I can't. Okay, next. So first marking point, oxygen bridge or glycosidic bond is broken. Show in your drawing, annotation. At the left-hand side of the chain, water is shown to be involved, or if you don't draw the water, at least you write that the process is called hydrolysis. So accept, A stands for accept. Number 4, free glucose molecules is being drawn, and then with the OH joined to carbon 1, and the chain now ends with the OH at carbon number 4. Okay, so maybe how to get the first marking point is when you show the working, you can annotate here in this process that the glycosidic bond is broken. So this is called annotation by the process called hydrolysis. So you can also write there by hydrolysis. Okay, then you can write here minus water molecule. Sorry, not minus. It's actually plus water molecule. Okay, plus water molecule. plus H2O. Okay, so with that, you will score the full marks. So we will stop here today. We will look at the cellulose molecule in the next lesson. So thank you.